Hello everyone, my name is Reshma Mehta and I'm a Principal Design Engineer at ARC Institute. Today we'll go over how to use Evo Designer, an interactive interface we created for DNA sequence generation and scoring using ARC's Evo 2 model. Evo 2 is a biological foundation model trained with 7 billion and 40 billion parameters on roughly 9 trillion base pairs from genome sequences across all domains of life. I'll start with the generation workflow, where you can prompt the model with a partial sequence or species and the model will generate new DNA. The generation prompt input works like a prompt in natural language AI tools you may be familiar with, such as Claude, Gemini, ChatGPT. But here, the prompt is a DNA sequence. You can adjust model settings in this panel, such as choosing between the 7 billion or 40 billion scale model and adjusting sample parameters such as the number of tokens, temperature, top K, and top P. These control randomness, diversity, and biological coherence in the generated sequence. Since Evo 2 predicts the next nucleotide based on the provided or previous context, these settings determine how the model selects from its probability distribution. For this demo, I'll stay with the 7 billion model and adjust the output tokens to 600. For the rest, I'll keep the defaults. There are a few ways to prompt the generation. The quickest one is to select one of the example sequences below. Clicking an example selects the organism and inserts 500 bases into the input field. Another option is to select or search for an organism from the provided set and generate a sequence with or without providing a starting sequence. The model receives the organism context to work from. For this demo, I'll go with the third option, which is to insert a partial sequence and setting the intended domain. I'm entering ATGC-TTGC-TT and asking the model to generate a prokaryotic completion. The input only accepts A, T, G, and C since this is a DNA sequence generator. The generation can take a moment to run. The result is rendered in a sequence viewer annotated with coding regions shown by this light purple bar that runs under the sequence. We also show these light green highlights on individual nucleotides to represent low entropy, conveying where the model is most confident or coherent with biological rules. The starting sequence that you supply is highlighted in blue. In this case, we supplied 10 bases, and the model came back with 610 base pairs since we had set the output tokens to 600 in the model settings. When you click on the purple bar, the corresponding protein structure loads in this ESM-Fold 3D viewer. You can also see the amino acid sequences in the open reading frames and select one to highlight the section for that segment in the structure. You can also study the score for this generated sequence in an alternate view. We will take a closer look at scoring a little bit later. Evo Designer offers some table stakes functionality. You can download the sequence as a FASTA file or copy it to a clipboard. Another cool integration is that you can blast a sequence to find known sequences in nature with functional similarities or relationships. Next, we're going to take a look at the actual scoring workflow. For this demo, I'm going to insert 1,847 nucleotides from the human actin beta example that's provided here. When you submit a sequence, the model will return a negative log likelihood score. This score is to indicate its confidence in the sequence. Lower scores mean higher confidence that the sequence is functional in nature. From a biologist's perspective, a negative log likelihood in the range of 0 to 1 is considered low, therefore suggesting that the model recognizes this is a real gene. Down here, we render a sequence logo plot. This is a visualization that biologists are very familiar with. It presents positional information. The height of the stack at each position indicates how conserved that position is. I can zoom in a little bit more so you can see the actual stacks and the heights for each individual position. Tall stacks are highly conserved regions that are functionally constrained. Mutations here would likely disrupt the gene function. I can use my scroll wheel to zoom into the plot and notice how the letters become visible. Look at the tallest letters in each of the stacks. You can see that these letters line up with the nucleotide in our submitted sequence. It is most clearly visible in these highly conserved regions. This alignment between the sequence logo prediction from the model and the submitted sequence presented below demonstrates that EVO2 has accurately learned the sequence patterns for this gene. You can also stack a per base entropy score visualization on top of the sequence logo. While the sequence logo shows what nucleotides are preferred at each position by way of presenting the probability of each letter in that position, the entropy clearly shows how strong that preference is, something that you can also tell in the sequence plot when viewing the high stack regions. Entropy and sequence logo are complementary views. For positions where entropy is less than 1, we color them green to draw attention to the highly conserved regions. The higher entropy regions appear in gray. So here we've actually scored a known sequence and we're able to check if the model recognizes the canonical pattern. As we mentioned earlier, it could also be interesting to score one of EVO's own generated sequences. The results would depend on the prompt flexibility and how much freedom EVO had to explore the sequence space, especially when generating novel sequences that don't mimic any known genes. And with this, I conclude this demo. Thank you for watching, and I hope this video helps you get started with EVO Designer.